a nice day. We can do it outside.
Oh, that's good. I can print it out and we can go out.
Should we read what it's saying for us to do? It's like directions on the function and so it's fun, right?
That would be for this. This is not recognized.
Yeah.
Ah, there it goes. Now why does it look? That's exciting.
You ready to do the interpretive dance version?
No, we're not going to do that.
I'll huddle around.
This is a new gadget that I just got.
And one of those things. Cool.
Yeah. How's the brewery coming along?
Good.
Okay. I'm going to need a beer really soon.
Yeah. 2
So-
weeks. Always 2 weeks from now.
Danny, can you ask Stefan for his little dongle?
I have one too.
Please. Why isn't this one working?
That's weird.
That is not. It's so weird.
Is it even plugged in?
And Jody was just using it.
The person going first.
How many tracks does it take to get
I
this
know,
straightened out?
right?
This is high.
It says computer 1, but computer 2 says it has
I think Michael's going.
It should be source 2. Source 2. No, it shouldn't.
Magic box. There's nothing.
Powering the computer.
Yeah. Look at that. Amazing what Stefan works and then one podcast doesn't. Oh, there it goes. All right. Where's your switcher changing? Plug that in. USB over on this side. All right, nothing like that very first of 18 talk bug that we now have fixed. Anyhow, I want to invite you, this is the first of 18 different talks. We have another one tomorrow. Madeline Duncan will be speaking about HIV at 4:30 tomorrow. And then on Friday, this same room, 3:10, we'll hear about anti-malarial compounds from Samantha Koski. And then we will hear about neurotransmitters from Lauren Lester Davis. Without further ado, I will turn it over to the presenter. Or introduce the introducer.
I was like,
right,
I'm not presenting.
Yeah,
Proteins Without Borders. It is my great pleasure to introduce Gus Lowry, who I first met as a freshman in chemistry who wanted to do some research. So before he ever came into my physical chemistry course, I said, oh, sure, let's do some research over the summer. I have some stuff that I'm going to be presenting at the national meeting, or the national ACS meeting in Boston. And I need a student to take a bunch of data with me, and then I'm going to hightail it to Boston. I was so impressed with his work that he earned first authorship. He pretty much went from just collecting the data to arguing with me on one of the deepest levels of interpreting the data. And I was just completely blown away. And that was when he was a first-year chemistry student. He then went on to the University of Twente. 2 years later? That's right, because he worked with Doug Martin in the physics department this following summer. Then you went to the Netherlands the summer after that. And then I believe you went to Oak Ridge National Laboratory. Is that correct?
Before that.
Before that?
Yeah.
So you can see that Gus himself has no borders when it comes to the science that he does.
Wow.
Anyway, it has been an absolute joy to have you in the research lab, in the classroom, and I'm excited for you to take over now as leading the classroom for us. So please welcome Gus.
Thank you everybody for coming, especially after Facebooking people and changing the time and emailing you all last night, particularly the professors in the back. Yeah, it's very nice of you to come. I am Gus Lowry, and I'm going to talk about some work that I did during the fall of my junior year. So that was 2016, and this was at Oak Ridge National Lab in Tennessee. So the project that I was working on was making a type of protein that makes cellulose, and that protein is called cellulose synthase, and it is a project for the Department of Energy. So the first question that most people would ask is, what do you do with cellulose? And the answer to that is everything. Cellulose is in probably all the clothes you're wearing. It's in most of our building materials, paper products, and animal feeds, and occasionally it ends up in human food as well. So this is the back of the Parmesan cheese container, if any of you are familiar with that. Cellulose powder is one of the ingredients to prevent caking. So yeah, occasionally you eat it. It's also used in drugs, and finally, biofuels. And so as far as the Department of Energy is concerned, this is the main thing that anybody cares about there. It's converting cellulose into biofuel. And so it's very abundant, and so that's the general idea, is that when we have an abundance of something, we can make use of it, and we can do that by making biofuels. So where do we find cellulose? Cellulose is in the is in the cell wall of a plant cell. So here's a nice introductory biology schematic of a plant cell. If you kind of blow up that cell wall, cellulose is this primary component, so it's this blue thing here. And that's what's giving it the structural rigidity and support that keeps plants upright. So that's why snap peas snap, I guess, is because of cellulose. So in all plants. So to make better use of cellulose, we have to know how it's made. And the thing that makes it is this protein, which sort of looks like a UFO, but it's not. It's actually a protein complex. And I know that it sort of looks like an Area 51 photo. That's a really great picture of it, though, I promise. So the currently accepted model for the cellulose synthase complex is that it's this rosette shape that's made up of 6 trimers. So that circle thing has 6 different lobes, and each of those lobes has 3 individual proteins. And so this is a schematic of that, constructed from the TEM image, or the SEM image there. And so each of those little dynamite sticks produces a strand of cellulose, and then those strands all are woven together and end up in the plant cell wall. And so in order to make better use of cellulose, we have to understand how it gets into the cell wall in the first place, so that we can pull it out easier and then convert it into biofuels. So looking at that individual component, this is the Synthase enzyme, or the cellulose synthase enzyme. And so this is the focus of the project, this particular protein. And like I said, this is another schematic. It's a 3D construction of the protein, and this is just what we think it looks like. We don't know for sure. This is the artist interpretation. And so basically what happens is the cellulose synthase protein takes glucose monomers, so these are little sugars, and it just stitches them together to make a cellulose chain. And then all of those cellulose chains come out and then are woven together and incorporated into the cell wall. And so, like I said, that process is not well understood. And one way to understand it is to make the protein and then structurally study it. So one of the questions that we have now is, how do we make a cellulose synthase protein? And the first thing to know about the protein is that it's a membrane protein. And membrane proteins come with lots and lots of fun challenges. The first being that there's multiple phases. And this is the primary problem with membrane proteins. So there's a hydrophobic region that crosses the membrane, the cell membrane, shown there, and then there's a hydrophilic region as well that interacts with the aqueous environment in the cytosol and then in the extracellular matrix. And so when you have 2 different phases, it can make it both difficult to make the protein and difficult to work with because they have such different properties. And so overcoming those challenges, we have to come up with some pretty creative solutions. So I'm just going to talk about I'm going to talk about a couple different ways to overcome that. So the first one is segmenting the protein. So I've given you this 3D schematic of what the protein looks like, but for the purposes of segmenting it, it's actually easier to think about it as a 2D representation. So these are just the same thing. I just sort of flattened it out and then made it in Microsoft PowerPoint. So that's the nice figure there. And so the segmenting approach is where we divide and conquer the protein. So we have these different regions that have like hydrophilic and hydrophobic interactions. And so if we segment it based on those, we can get all of the hydrophilic regions by themselves, so the N-terminus and the catalytic domain. That's what those 2 regions are named. And then we can get the hydrophobic domains by themselves, and that's those transmembrane domains. And so this approach to segmenting the protein makes it easier to work with, because then we can control the number of phases that we have when we're making the protein. So. Yeah, so this is some intro bio for the physicists in the back of the room, particularly John Brandenburger. Oh, hi, I didn't see you there also, sorry about that. Yeah, so the central dogma of biology is that you can turn DNA into proteins by transcribing into RNA and then translating the RNA into an amino acid chain, which ultimately folds into a protein. So if the goal is to segment a protein, what we actually do is segment DNA. And so that's just something to keep in mind, that instead of making a protein and cutting up into pieces, we're actually cutting the DNA up into pieces and turning it into a protein. And so that's what we'll do with this approach to make sessile proteins. So generally, structural biologists make proteins with E. coli, and E. coli is a bacteria. And these bacteria are like little molecular factories. So we give them the DNA instructions to make a protein, and then they just sort of crank out a protein if we induce them to do it. And so this has worked really well for the lab group that I was a part of. They've made the N-terminus domain, or the N-terminal domain of the protein. And so this is the ultimate goal right here. These are structural conformations that describe, or that show what the protein looks like. And so these are possible arrangements that that the protein amino acids can fold into. And so this is just for that N-terminus because it's hydrophilic. It's easy to make with E. coli, and they've also, the lab group has also made the catalytic domain, which is another hydrophilic region of the protein, but with less success. So this approach can work really well to have E. coli make proteins for us. But one of the challenges is with membrane proteins, these transmembrane domains can actually kill the E. coli. So before they start making the transmembrane protein, but then once they've made enough of it, it kills them, and it's they usually haven't made enough of it for us to study it. And so in order to make membrane proteins or transmembrane domains, we have to come up with a different approach to that. So that approach was what the basis of my project was. So this is making transmembrane domains with cell-free synthesis. And so with the E. coli, we couldn't make these proteins because they would die. And so if we go cell-free and not use any cells, then we don't have anything to kill. And so we might be a little more successful with making the proteins. So cell-free synthesis, like I said, is making a protein without cells. And it just has 3 inputs. And I swear you actually put it into a test tube. That's not just like a mixing pot analogy. So the first thing is that you need to have the genetic information for the protein. So like I said, that segment of DNA that translates into the protein. And then you also have to have the cellular machinery necessary to express a protein. And so that would be things like ribosomes and polymerases and cofactors. And then finally, you add small molecules that are needed to operate cellular machinery. So that would be things like ions and cellular energy, and then the building blocks for proteins and DNA as well. And so when you mix all 3 of those things together, the volumes that I was using were about the size of a teardrop, or maybe 1/10 of a teardrop or something like that. So these are really, really small volumes. And I would have brought in some teardrops today from Graham's exam, but I didn't bring my test tubes. But anyway, yeah, so anyway, when you mix all these things into this test tube and you get this little teardrop volume, you just incubate it for like 2 hours and it makes a protein. And the fact that that's a spontaneous process is just amazing to me, that I was able to make a protein in a teardrop size volume in 2 hours. Like that to me was the craziest part about this. And it's actually very adaptable, and it's an excellent way to make membrane proteins. And so the adaptability is just that, like, you can change the ratios of things that you're adding, and you can also add additional things that are, I don't know, maybe might help membrane protein expression. So this is an excellent alternative to E. coli when that doesn't work. So this is a general overview of my workflow at the lab. So like I said, the first thing was making these DNA plasmids that describe the protein, then translating them into proteins with cell-free synthesis. And then these 2 things are sort of first checks, or just making sure that you made the correct protein. So the first one, SDS page, is like the initial, like, did something happen, and then Western blots tell you whether or not it was the correct protein that you made. So I'll talk a little bit about those soon. And once you confirm that you made the correct protein, then you can scale up the reactions to go from teardrop to maybe like 10 or 15 teardrops, solubilize the protein, and isolate it by separating it from all the other things that are in the test tube. And then once you have the protein by itself, you can do structural analysis. So that would be things like X-ray diffraction or neutron scattering. And that would get you those structural models that I showed with the N-terminus a few slides back. So that's the ultimate goal, is to get structural data for this for these transmembrane domains. So SDS page, like I said, is this sort of first check to make sure that you made the correct protein, or that you made a protein in the first place. And the way that it works is it's separating proteins via an applied electric field. So SDS is an acronym for sodium dodecyl sulfate, which is a detergent. And so that detergent gives all of the proteins in a protein mixture a negative charge. And then when you put it in an applied electric field, that's the page part, it's polyacrylamide gel electrophoresis. You put the protein mixture into a gel matrix, and then the negatively charged proteins will move towards the positive pole, and then that ultimately stratifies the protein mixtures by their apparent molecular weight or size generally. So smaller proteins will end up at the bottom, larger proteins stay at the top because they just don't move as fast through that gel matrix. And so this helps assess the reaction success, like I said, but then we also have to confirm that it was the correct protein that we made. And so the way that we do that is with a Western blot. And this is another SDS page gel here, so just not to fool anybody. But yeah, so this is a highly sensitive way to tell that you made the correct protein. And what we do is, there's some nice molecular biology tricks for this, and in particular, we put a component of the protein, so we add this thing that's like an unnatural element, something that wouldn't be in a protein from nature. And that element is what we're looking for with a Western blot, and that's called a his tag. And so it's just this, it's a combination of amino acids that would never occur in nature. And so when it does, or when we are looking for it, the protein that we tried to make is probably the only one that's going to have it. And so we take an SDS page gel, and then we apply antibodies that are specific to that histidine tag. And then ultimately, it's like we're asking the question, is the protein there? And then we see the Western blot, and it says yes, because there's a little band for that protein. And so it's just saying that this protein has that histidine tag, and that tells us that it is the correct one. So that's a nice way to check to make sure that you made the correct protein, and then you can scale up the reactions for the cell-free synthesis. So just to go through some of the data, the first protein that I tried to make was the first 322 amino acids, and so that's the N-terminus and the first transmembrane domain circled here. And so this is, I already showed you this data, surprisingly. That protein is shown there, and then the Western blot to confirm that I made the correct one. For the second transmembrane domain, I tried to make the 814th through the 1065th amino acid, and so that's that region there. And that is shown on the SDS page gel here. And this is just the orient for the Western blot. And so that's also shown there. And so these are just making sure that I'm making the correct protein. And then from there, I can scale up the reactions and try to isolate the protein to do structural analysis. So at the current state, making this protein, you just have a mixture of proteins. So it's this protein and then like, I don't know, maybe 100 other proteins in that mixture. And so to try to get the protein of interest alone by itself, we have to do another separation technique. And that technique was called size exclusion chromatography, or SEC. And so size exclusion chromatography takes a protein mixture, like the one shown here, pink big curls and small pink curls, so typical proteins. And so it runs it through this column, and the column is packed with these little spheres that have pores in them, and then the pores will take in the smaller proteins, and the larger ones will pass through, if that makes sense. So this is the SEC column, and things are coming off of the column, so they like drip through the column, and then we collect little fractions, like little test tubes of the stuff that's coming off the column, and then we can analyze that to see which one has the protein we're looking for. So what you end up with is a bunch of fractions, and that's what we call them, cell fractions, and then ideally the protein that we're looking for, maybe this one, would be alone in a fraction. And so this is, so the larger proteins come off first, and then the smaller ones come off later because they were stuck in those little pores, the nice green pores here. So this is what an SEC trace usually looks like, or a plot looks like. And so on the y-axis we have absorbance. Essentially what that is telling you whether or not an event has occurred. So every time there's a little bump here that's like, something happened. And you want to collect fractions from that. And so as we go across on the x-axis here, that's the eluted volume. And so at each point, like maybe like every little bit here, we're taking a fraction. So we might take a fraction here, and a fraction here, and a fraction here. And those are just little volumes that hopefully contain the protein that we're trying to separate out. And so then what we do with those fractions is we run them on SDS page to try to see where our protein is, essentially. And so this protein here is the protein that I was trying to separate from the additional proteins shown there. This is for the 1 to 322. And so, yeah, so it's a mixture of proteins. We've got the nice protein we want and then some other ones that we don't want, and we're just trying to get rid of those. And so I put that protein mixture shown in this column, or in this lane, I put that onto the SEC column, and then picked up fractions, which are the next few lanes. So each of these lanes represents a fraction, and then I will correlate them with the peaks. So the first peak here was not the one that we were looking for. That's good. The next peak, which looked very promising because it's so prominent, surprisingly not the protein we were looking for. And then the next peak, this like sort of little humble one, seems to be the right protein. And then additional products that are of similar size came off right after that. And then finally, this last one we don't care about, but another thing that came off the column. And so this is a nice way to separate the protein mixture. So before we had a mixture of lots of proteins, and now we have more purified protein. And from that purified protein, we can do structural analysis. So I'll talk a little bit about one of those methods. So the primary one that I did was circular dichroism, and so this is just something that reveals basic structural features of a protein sample. This is called CD, and so CD produces a plot that looks like this, and I know that this plot looks really uninteresting, but it has a very key feature that is characteristic of something common in transmembrane domains, and it's a little bump here, and then a little bump there, which I'm sure everyone can see very clearly. This made me very happy. So anyway, so characteristic of an alpha helix, which is common in transmembrane domains. And so this is a confirmation that that 1 to 322 sample that I purified is the correct protein, which is excellent news. So in summary, I use cell-free synthesis to produce 2 different protein segments. The first one containing the N-terminus and the first transmembrane domain, and the second one containing the second transmembrane domain of the Synthase protein. And previously, these had not been made with E. coli or any other methods. So this was pretty exciting to come in and use a technique that I'd never used before to make these proteins. And then I was able to separate the Synthase 1 to 322, so that's this protein segment here, using size exclusion chromatography. That's like a nice reminder of what it looked like if you forgot 2 slides ago. And then finally, it showed that that segment had alpha helical secondary structure. And this is an alpha helix for anybody who's wondering. But yeah, so this was just confirming that it was the correct protein in the first place. So the future directions would be to make more of it. That's always a struggle with cell-free synthesis. It's a very expensive technique to do. That's why I was doing teardrop volumes, because they're very expensive reactions. And so in order to make more, you'd have to scale up the reactions quite a bit. And so that is a cost challenge. And so to overcome that, the group is going to try some other methods as well. One of them being eukaryotic cell expression. So bacteria are prokaryotes, and the Synthase protein is a eukaryotic membrane protein. So expressing that protein in eukaryotic cells may actually be easier than trying to do it in bacteria. And particularly insect cells, I guess, are very nice for membrane protein expression. And I Googled worried insect, and that's what came up. I think it's from a children's TV show. But yeah, so it's a promising method for expressing membrane proteins. And so that is the future directions for the lab group. I would like to thank quite a few people, actually. So Chu Zhang and Hugh O'Neill were my lab mentors at Oak Ridge National Lab. Vanu Vandavasi was a postdoc in the group who is now at Vanderbilt. And Kevin Weiss is the lab space manager, or is the lab space manager there. Julie Malakote and Cheryl Brummett were 2 people who are in the administrative side of things through a program called ORAW. Craig Teague was the resident director during the program. And Dan Gibson and Beth Jeffries are both from Denison University and did the ACM side of things when I was down there through the program. And then finally, I'd like to thank people from Lawrence, including Allison Fleshman, who introduced me and is my life advisor and coach, Doug Martin, who was my second life advisor and life coach, but who could not be here today, unfortunately. Graham and Dave for the senior experience class. Dan Martin, of course, because of doing everything for me all the time. It's amazing. Thank you. I'm so sorry about all of it. And then finally, my contemporaries, which I think is a funny way to say my classmates, which I guess you're over here on this side of the room. So thank you. And then funding sources. So I'm especially grateful for the Zelke grant and the Senior Experience Fund, which helped me do some cool things this summer and then this fall. And then funding through the ACM and ORISE made sure that I had food to eat during the program. And then all of the stuff at Oak Ridge was paid for through a grant from the Center for Lignocellulose Structure and Formation, which is a DOE grant that my mentor had. So, yeah. Thank you for your attention, and if you have questions, nobody has questions? Graham, please.
So if you're giving specific plasmids in cell-free method, why do you get all these other proteins showing up in your gels?
Because all those other proteins are used for the protein expression. So things like cofactors or proteins that help polymerases bond, and so all of those proteins would be in that mixture. And so, yeah, there's lots and lots of proteins, and then you're just looking for the change in protein, just the one extra one. So, yeah.
Follow-up question to Graham.
Yeah.
What was the source of the ribosomes?
Like what organism?
So you have wheat germ extract,
Yeah.
rabbit reticulocyte extract, the list goes on.
I think all of these were prokaryotic. I think they were all E. coli ribosomes, I'm pretty sure. Does that not sound right? I think it was. I'm almost certain. So, yeah, I mean, these are all cell lysates from E. coli that we used for the cell-free expression. So some of them are recombinant kits, and the other ones are just lysates.
So did you have to make the RNA, and then so it was a transcription and translation combination?
Yeah, and it was all downhill. It was like you put it in a test tube and put it in the incubator, and then it was a protein. So pretty sweet. Dan?
How do you confirm that it folded the same way that it does in the intercellular membrane?
Yeah, that's a really good question, and I think that you have to get SANS, or like a small angle neutron scattering data to confirm that it did. So you need like data from multiple sources to really tell you. Like it's a combination of lots and lots of things to say, like, is the protein folded correctly? And I mean, part of solubilizing it is putting it in detergents, and I mean, hopefully that's not changing the properties of it or how it's folding, but I think the short answer is it does. So that's part of the challenge, I guess. But
I think I missed this at the beginning. The molecules are so much bigger than I'm used to using.
What? And the proteins?
They're huge.
Yeah.
They're so big. Anyway, so you talked about DOE's real interest is biofuel and stuff. And so can you connect back? I mean, the poor little insect at the end was kind of upset about possibly what's going to happen to that little thing.
Yeah.
How is that going to relate back to biofuels, and where does your work fall into that progress forward?
So it's mostly just figuring out how cellulose is incorporated into the plant cell wall. It's super difficult to take it out. It's cellulose is found in the cell wall in combination with something called hemicellulose, which is as related to cellulose as it sounds, and then lignin, which is another component. And so those three things kind of come together and are very difficult to separate out. And so when they're trying to make biofuels, they just need pure cellulose. And so there's a lot of different ways to go about that. So some of it is like genetic engineering of plants to try to get more pure versions. Part of it is just wondering how it gets in there. And that's this component of the project, is figuring out how the protein functions and how that function turns into cellulose being in the plant cell wall. Michael?
Can you, I may have missed it, can you talk a little bit more about your reasoning for segmenting it the way you did, starting with the first segmentation?
Yeah, so the segmenting is like, so if you try to express the full protein, it doesn't work in E. coli because the protein has too many transmembrane domains and it kills the cells. And so breaking it up into smaller bits, you can sometimes have success expressing it in E. coli, but it's just generally easier to express certain sizes of proteins. So like much larger proteins, you tend to have things get screwed up. And so just cutting it down to smaller parts is one major way to do it. And also the phase difference is another challenge. So the reason why the 1 to 322 worked is because there's only 2 transmembrane domains, and I think that makes it less difficult to handle. So.
Is it all expressed in the same chromosomal location?
Sorry?
Are all the segments of the entire complex expressed in the same location?
Like in plant cells?
Yeah, like on the chromosome.
Yeah, so in plant cells it would just be one gene.
Okay.
But then when we do it in E. coli, they're just plasmids.
Can you give any sense of time scale and cost between the cell-free method and the traditional method?
Well, the cost is, so E. coli are really cheap and disposable, and it's all very, very easy. So, I mean, this is sort of like a last-ditch effort to do an alternative method. Really, they've tried to express all of the different, they've chopped up the protein into every single thing you can imagine and tried to express it in E. coli, and it hasn't worked the majority of the time. They've had some success, but really, like the lab group has had some success, but really it's been challenging. And so this was like a last-ditch effort, and that's why they were willing to pay that much. So it's nice to know that you can do it and that this is an alternative to doing it, but I think they're going to look into eukaryotic cells, which I think are cheaper as well. Definitely much more like technical, but cheaper. So yeah. So there's a postdoc right now who's working on that. So, other questions?